All right. So it's my great pleasure to introduce Panos, who's originally a chemist, originally from Greece. He did his bachelor's and his master's degrees in chemistry at the University of Patras and a PhD in chemical engineering at Manchester. He's currently a research fellow in computer science and creative tech at the University of Western England in collaboration with and in the group of Andy Adamansky, a very dear friend of mine. And Panos is interested in proteinoids. In fact, that's what he's going to talk about today, and their potential applications in unconventional computing, and in particular, the synthesis of proteinoids under conditions that are interesting, and as well as exploring their electrical properties. I won't say any more because that's actually the topic of his presentation. So when you're ready, don't feel like you need to sing it, but feel free to if you sound like you have a nice voice. Fire away.
Okay. Hello everyone. My name is Panos and I'm a research fellow in the Department of Computer Science and Creative Technology at the University of West of England. Today I will present our latest work on developing neuromorphic computers using synthetic protocells, which called proteinoids. Neuromorphic computing is an exciting field that aims to mimic the structure and the function of the human brain for computational purposes. Traditional computers separate memory and processing, whereas brains compute and store information in the same components, neurons and synapses. A key challenge is to find the suitable materials to imitate neurons and synapses. Our work looks at primitive cells that we call them proteinoids or thermal proteins. These kind of compounds were first synthesized by Fox in the 1960s, inspired by the work of Oparin, who suggested that the theory of origin of life as conservates. And before that, on key foundation of Aristotle, the life as primitive as a life force. The proteinoids are designed to incorporate memristive self-assembling proteins that can modulate conductivity in response of light, especially acting as neural like cells. Protocells are vesicles made of lipids, proteins, and nucleic acids that can display lifelike behavior such as metabolism, movement, response to light. In this slide, you can see a scanning electron microscopy of proteinoid microsphere synthesized in our lab. The presentation will cover our latest findings on the single integration capabilities of proteinoids and progress towards multi-compartment neuronal mimics. Transitional to the next slide, let's dive deeper into proteinoids. These slides compares the electrical impulses recorded from crayfish neurons to those measured in our proteinoids microspheres. On the left is a spike that trained from crayfish sensory neuron, showing in rapid polarizations of 20 millivolts, lasting around 10 milliseconds. This matches remarkable well with the spikes exhibited by our synthetic proteinoids on the right. Yet, despite their simplicity, the proteinoids display sophisticated electrical properties akin to neurons. Like neurons, the proteinoids generate potential and can integrate multiple signals arriving in quick successions. This provides evidence that proteinoids have an innate neuron-like excitability. The electrical signaling may arise from conformational changes in proteins and diffusion of ions across the microsphere membrane. This slide highlights the exciting electrical behaviors exhibited by proteinoids microspheres. On the left is data from Mishima et al. Showing in rhythmic burst of endogenous activity from proteinoids made of thermal polyamino acids. The right figure from Przybylski, bottom picture from Przybylski, demonstrates sensory responsiveness in a proteinoid sphere composed of polyaspartame, glutamate, and arginine. Here we see trains of electrical discharges induced by pulses of light stimulation. The proteinoids transduce the light signal into regulate spikes akin to a photosensitive cell. Together, these features reveal that proteinoids have an innate capacity for complex patterns of electrical signaling, including oscillatory rhythms and sensory integration. Neuromorphic computing seeks to emulate the architectures and capabilities of the brain through hardware and algorithms. As the growth chart shows, research interest in neuromorphic computing has rapidly accelerated in recent years. Key features of neuromorphic computing include massive parallel processing, collocating computation, and memory as in neural networks, and adaptive learning capabilities. Neuromorphic computing designs are enabled by memristive devices, which can mimic synapses along with spiking neural network topology and brain-like learning. Major benefits include efficient pattern recognition for machine learning, low power consumption, and high scalability and fault tolerance. This makes neuromorphic computing ideal for applications like image recognition, natural language processing, autonomous robotics, and medical diagnostics. Now turning to the next slide, the proteinoids exhibit compelling electrical signaling behaviors that parallel neuromorphic systems. Recent research demonstrates that proteinoid assembles can propagate spike-like potential and spike-like potential. The proteinoids can act as couple electronic elements that can transmit signals down to the chains, analogous to the action potential in exons. The signal frequencies depend on factors like the microsphere composition, the surrounding environmental conditions. This shows the potential for using the proteinoid excitability. Overall, this discovery of electrical signaling in proteinoid networks is highly promising for constructing synthetic bioinspired information processing systems. The protonic conduction provides a mechanism for transmitting signals between proteinoid neurons. Also, research provides compelling evidence that proteinoid systems can process information by electrical inputs. By applying electrical pulses to proteinoid microspheres, scientists measure the relationship between the input signals and the resulting outputs. The proteinoids demonstrate signal amplification effects as well as integration of multiple inputs over time. The input-output processing resembles that of a transistor circuit. The transfer functions arise from reconfiguration of the proteinoids microspheres in response to electrical inputs. Harnessing the emerging properties of proteinoid materials will enable future biocomputers based on artificial bioneural principles. In the next part of the talk, I will expand on potential architectures and applications for proteinoids computing devices that leverage these information processing capabilities. Moving to this slide, our work has uncovered interesting light-responsive behaviors in proteinoids that could be harnessed for optogenetic style control. We found that cold white light triggers electrical oscillations in proteinoid ensembles. The amplitude of strength of the oscillations decreases under continuous illumination, as you can see in this plot. However, passing the light on and off induces stronger, more pronounced oscillations compared to constant light. This shows that proteinoids can sense changes in lighting, not just intensity. The transient spikes implied proteinoids may have an internal clock-like oscillator that activates with light pulses. Under constant illumination, the amplitude drops, but the oscillation frequency rises. Overall, this demonstrates the potential to optical control the emergent electrical properties of proteinoids. The light-sensitive excitation could enable optogenetic neural-like networks using only proteinoids and light stimuli. Much remains unknown regarding the photosensitive mechanisms in proteinoids that produce these oscillations. In the next part of the talk, I will discuss the implications of these photoresponsive behaviors for engineering optical controlled proteinoid computing systems. As we have seen, proteinoids display rich electrical signaling behaviors, exciting new research shows that this can be harnessed to implement Boolean logic operation using proteinoid systems by applying specific patterns of electrical pulses as inputs and detecting resulting spike patterns as outputs. Proteinoids can realize key gates, logic gates like AND, OR, and NOT. The input pulse combinations are mapped to proteinoid spike outputs based on their timing. This spike timing encodes the logic result. For example, two pulses may trigger a spike, while one pulse does not, implementing a net gate. The presence of absence of spikes in response to the input pattern determines the logic output. This provides a proof of concept for using proteinoids as synthetic biology logic elements. In the conclusion, I will expand on how proteinoids could enable example shift to green analog bioelectronic computers that operate via synthetic principles. This brings us to an exciting application of harnessing proteinoids for speech recognition. Our research shows that proteinoid assemblies can recognize spoken letters. In these experiments, sounds of the English alphabet were converted into electrical waveforms and fed as inputs to proteinoid solutions. The electrical responses were recorded and analyzed. The proteinoid spike patterns were less regular compared to the audio input signals. They exhibit a lower dominant frequency and wider distribution of amplitudes. Remarkable, we found each spoken letter of the alphabet mapped to a unique proteinoid response pattern. This demonstrates the potential for proteinoids to discriminate voices. While the mechanism remains unknown, this early results provides a proof of concept for speech recognition with proteinoid systems. Their tunable electrical responses enable audio waveform discrimination. With further optimization, engineered proteinoid materials could potentially serve as the basis for biocompatible speech recognition devices. Synthetic bioneural computing could transform how we interact with and use technology. Now I will go to the next slide by summarizing key challenges and opportunities in making functional bioelectronic computers from these synthetic protocells. These features provide an initial framework for investigate communication between interconnected proteinoids by drawing analogies to neural systems. In biological neurons, presynaptic and postsynaptic cells are linked by synapses. Neurotransmitters transmit across the synapse can induce an electrical response in the receiving neuron. While proteinoids exhibit spike-like activities, it remains unknown if similar synaptic style signaling occurs between proteinoid microspheres. This model represents an abstract simulation of potential signaling in a proteinoid network using computer metrics like PSI and PPI to reflect their electrical behaviors. However, this feature does not confirm proteinoids can actually mimic synaptic functions. Further chemical and electrical analysis is needed to validate if proteinoids demonstrate true neuronal-like communication. Uncovering signal pathways in proteinoid networks could suggest ways of engineering them as synthetic neural systems for information processing and collective behaviors. This slide compares some of the similarities and differences observed so far between proteinoids and biological neural systems. On the one hand, proteinoid signaling exhibits spike train-like responses analogous to neural action potentials. Proteinoid networks also demonstrate characteristics of neural collectives like plasticity, learning, and primitive information processing. However, there are also fundamental differences. Individual proteinoid elements are intrinsically far simpler than neurons. The mechanisms for interaction between proteinoids remain unclear and may not fully recreate synaptic communication. Additionally, the architecture of proteinoid networks follows more stochastic self-assembly rather than genetically guided optimization. Overall, side-by-side comparison to natural neural systems provide a conceptual framework for elucidating the origin of adaptive cognition in these synthetic protocells, while many open questions remain regarding how collective intelligence emerges in proteinoid materials. In the conclusion, I will summarize the outlook and opportunities for further unlocking the inner workings of this compelling form of synthetic biological intelligence. Previous slides demonstrate the exciting electrical signaling behaviors of proteinoids, from endogenous busting to sensory responses. This neuronal-like excitability makes proteinoids promising candidates for bioinspiring computing. A key advantage is that the proteinoids exhibit innate biological computing abilities like integration, adaptation, and short-term memory. Their dynamics arise from charge distributions and conformational changes in constituent proteins. With careful molecular design, we can tune proteinoids to mimic neural plasticity for learning. However, significant challenges remain inherent in proteinoids for computing. We need improved techniques to construct organized networks of proteinoids that can process information across multiple compartment and length scales. To explore the computing potential of proteinoids, we have developed models of proteinoids ensembles using reservoir computing techniques. Reservoir computing is a framework for training recurrent neural networks to perform tasks like pattern recognition. Shown here are simulated configurations of proteinoids interacting with an electron array. The proteinoids are represented by the black spheres, surrounded by the red electron grid. This mimics the experimental setups where we apply electric fields and record responses across a population of proteinoids in solution. In our reservoir computing models, each individual proteinoid is modeled as a node with adaptable synaptic weights and fire dynamics based on empirical proteinoid measurements. The population forms recurrent networks whose complex dynamics serve as a reservoir of computing power for learning. The reservoir computing example is well suited for harnessing the proteinoid's intrinsic computation abilities for unconventional computing application. Our modeling work.
But sorry to interrupt. You're doing really well, and we're not running. We're on time. But because I want there to be, it's a very interesting talk, and I want there to be a couple of minutes for questions. I'll maybe if we wrap up in a couple of minutes. Yeah.
So I will continue.
Yeah, but if you just wrap it up in the next couple of minutes, so we could have time for questions.
Okay.
And then there's a coffee break for everybody here who is like,
okay,
their glucose is collapsing.
Okay. This slide shows simulate electrical readings from a 16 electron array interacting with proteinoid reservoir model. The reservoir was simulated with different binary input patterns represented by pulse on electrodes E9 and E3. Input 10 generates spikes only on E9, while 01 stimulates just E3. The double impulse of 11 activate both sides. The ability to generate complex but decodable outputs in response to simple inputs highlight how proteinoids reservoir can perform usual transformations. This slide summarizes the variety of logic gates emergently performed by proteinoid reservoir model. The table lists how many gates of each type were decoded at every electrode in the 60 channels array. We see diverse logic functions arising across a network when driven by different input patterns. Some electrodes, like E1 and E16, show NOR gates with no spiking for either input. Meanwhile, electrode E8 demonstrate both OR and AND logic in response to various stimuli. Rather than designing for specific gates, the network inherently produces diverse temporal dynamics that we harness for different operations. Understanding and controlling the complexity of the proteinoid system remains an ongoing challenge. However, these results emphasize the exciting possibilities for training proteinoid networks to perform unconventional bioinspired computation. Finally, this slide outlines the concept of using proteinoids as unconventional computing substrates. Each proteinoid have shown basic logic operation, pattern recognition, and neuromorphic-like phenomena. However, key hurdles include scaling up device complexity, developing biocompatible interface in a packaging, and ensuring stability of proteinoid materials over long-term operation. The future outlook is a revolutionary paradigm shift to bioinspired computing, where green adaptive intelligent processors are grown rather than manufactured. Realizing this grass version will require decoding this mechanism of emergent proteinoid cognition and harnessing the collectives for engineering desirable functional behaviors. Thank you very much.